thank you to all of our partners. So it gives me really great pleasure to introduce you Dr. Ram Akalish, who's a clinician scientist and an associate professor at the University of Washington in Seattle. Was it not the long Thanksgiving weekend, I think he would have come up to Vancouver to visit us, and he will another time, but has agreed to do this for us. He received his MD and PhD degrees at Washington University in St. Louis, and he's a board-certified anatomic pathologist with specialization in kidney pathology. He's internationally recognized as an expert in genomic technologies applied to human kidney tissues, including spatial transcriptomics. And he's received awards for his research, including the Gloria Gallo Research Award from the Renal Pathology Society and the Young Investigator Award at the 14th International Podocyte Conference. And so really it's a pleasure and a privilege to be able to introduce him, but actually more excitingly to listen to what we can learn from kidney biopsies. So over to you, Ram, and thank you so much for making this on a holiday Friday for us.
No problem. Thank you. All right. Is the screen presenting all right? And you can hear me?
Yeah, perfect. Thank you.
Great. Thank you. Thank you, Adira, again for that nice introduction. And thank you again for the invitation to come speak with you this morning. As Adira said, I wish I could have been there in person, but taking some time to spend it with family here at the Thanksgiving holiday here in the United States. So today, really what I wanted to describe to you is some of the new technologies that have been emerging on the scene in the last 2 to 3 years. As a pathologist, I think about, how we can extract the maximum amount of information when a kidney patient undergoes a biopsy, an invasive procedure that extracts some of the tissue. I feel we're leaving a lot on the table currently, but there are new technologies that may help us extract more information that may guide either our diagnostic algorithms, or even shed light into how the patient's disease course may progress and suggest novel interventions. And so that's really kind of the goal I wanted to describe to you today, and describe some of our efforts towards achieving that. So I have nothing relevant to disclose for today's talk. So as I shared before, the main learning objectives for this talk is to kind of understand a little bit about the principles of what I mean by spatially localized gene expression profiling, or spatial transcriptomics. This goes by a variety of terms in the literature, sometimes with the two main commercial platforms labels attached to them. But I'll describe this to you, and hopefully you'll have some sense of what that means by the end of the talk. The bulk of the presentation is going to be thinking about how we can extract clinically relevant insights using these technologies. And I will present to you a variety of studies, some published, some unpublished, that we have done using kidney biopsy tissues and kidney tissues in general to learn more about the underlying biology. And finally, I'll interweave within this some considerations and challenges. With any new technology, it's very easy to get caught up with the hype and get super excited about the promise of them. And while that's good and can motivate us, we also have to be careful and keep an eye on the limitations and blind spots that these various technologies have, so that we don't overinterpret or misuse them in the wrong context. So let's begin with kind of an outline of what I mean by spatial transcriptomics. And so for this, I'm going to actually go back in time a little bit and think about the beginning of microscopy. So this is Anton von Leeuwenhoek in the 1600s. And on the left, you can see the microscope that he created, which doesn't look like any kind of modern microscope we've seen. But here was a little lens. This little screw-like device would hold a little bit of a sample or a material at the very end of its pointer. And he could move this back and forth or up and down. And then by looking on the other side, you could see a magnification of whatever was being held there. Now, even though this looks like a very primitive device, Leeuwenhoek's secret sauce, if you will, was actually creating this lens. And he was he guarded that secret very, very closely. And also in Italy, lens makers and microscope makers telescope makers were also very interested in creating microscope-like devices around the same time as well. But Leeuwenhoek's real advance, besides making this microscope, was also using some early staining techniques. And with that, he could actually get contrast with what would have otherwise been very transparent materials. So with that, and with this kind of microscope, he was able to study all of these different microscopic structures and creatures and parts of the body, which was really quite amazing. And again, we didn't have photography then, so he had to actually draw this all by hand and color it, which is also kind of a remarkable feat if you think about it. So since then, we've progressed quite dramatically in terms of our ability to extract a lot of information of the tissue. And this, of course, forms the principle of pathology laboratories around the world. So the general process, of course, is we take a piece of tissue, say a kidney or kidney biopsy. This is then fixed in formalin. So this is a material, of course, which cross-links all the proteins so that it preserves the structure and prevents it from decaying. It also imposes some rigidity to it so that it can be sectioned properly for the microscopic evaluation. That material is then embedded into wax-like substance called paraffin. So this whole thing is sometimes abbreviated as FFPE, for formalin fixed paraffin embedded. And then thin sections, about 2 to 5 microns in thickness, so are applied to glass slides and then stained. So just to think about that thickness of 2 to 5 microns, that's thinner than the width of a single red blood cell. That's the volume of tissue that we actually examine on each of these level sections of the microscope slide. And so for hundreds of years since Leeuwenhoek's time, we could use a variety of different stains, which would give us different color palettes, by highlighting different biochemical properties or structures of the tissue. And this became very useful. For example, we could highlight fungal organisms in this GMS stain. We could highlight basement membrane-like structures in this Jones stain, which we use all the time in evaluating kidney biopsies. We can highlight mucus with Alcian blue, for example, and so on and so forth. So these different stains really were leveraging the biochemical properties to highlight different structures, provide contrast, and give us additional information of what might be going on within the tissue. And this was really the state of pathology for about 400 years. And still very useful and productive in terms of classifying and identifying disease. Now, another way to extract additional information from the tissue is to maybe think outside the usual light microscope and use a different microscope altogether. And so, for example, in evaluating kidney biopsies, we routinely use electron microscopy. So this requires a separate portion of tissue to be submitted in a glutaraldehyde fixative, a slightly different fixative. And then it's post-stained and examined at very high magnification using an electron microscope. This is, of course, we use this in kidney all the time to think about where immune deposits might be, whether there are amyloid fibrils, to examine the integrity of the basement membranes and podocyte foot processes, as an example. So that's an example of a different kind of microscope that has entered routine clinical use. However, there are a variety of other advanced microscopes which also give us a different view of the tissue. So some of these are label free, such as like Raman scattering microscopy, which again uses biochemical properties of the tissue to provide contrast. Mass spectrometry imaging, where heavy metal labeled, isotope labeled antibodies can be used to depict and identify different parts of the structure or even different lipid composition of the tissue. And of course, we can even take the whole 3D reconstruction and visualize it using light sheet microscopy approaches. But all of these require specialized sample processing workflows, which may not fit very well into a routine clinical laboratory. So that's kind of the downside of these different microscopes. A third way to extract different kinds of features and information is to use different stains. And this is, again, techniques that we use routinely in the clinical laboratory. We use immunofluorescence and immunohistochemistry, which are antibody-based detection methods. So we can use an antibody that detects a particular protein or antigen, and then use either fluorescent labels or a chemical chromogen deposition to identify the site where that antibody is bound. We can also detect RNA molecules, so different transcripts or viruses using a similar approach called in situ hybridization. And here this is shown in a red stain in this particular glomerulus. I know colleagues are also exploring the possibility of using artificial intelligence or deep learning approaches to extract, not only segment different features of the biopsy, and what I mean by that is identifying different structures, but actually quantify and extract mathematical information about the size, the volume, the relative proportion of different cell types within the tissue, and therefore correlate that with the pathology diagnosis. So this is, again, an evolving field that we will see more of in the coming years. And finally, another way to extract more information is to increase the plex. And what I mean by that is to increase the number of analytes that are simultaneously labeled and measured in a single tissue section. And so again, there are a variety of commercial approaches that are doing this, either to detect proteins or other antigens within the tissue. We can also multiplex in situ hybridization for detection of RNAs. At simultaneous detection, it's possible to go up to about 8 or 12. But really, to go much higher than that, we have to take advantage of sequencing-based approaches. And that is really where spatial transcriptomics comes into play. Instead of using an actual physical label or a dye to read out the presence of a single RNA molecule in its space in the tissue section, we use the sequencer as a microscope to read out the entire transcriptome and to localize it within the tissue. Now, I wanted to just step back and bring this concept home as well as to why we would want to have spatially registered gene expression profiles. And so many of you might be familiar with RNA-seq. So this is a sequencing-based approach to quantify the level of expression of all genes in our genome, for example. And this was developed initially in a bulk method, meaning that an entire tissue or structure had to be broken down, its RNA extracted, and then quantified by sequencing. Now, this is very useful and still provides a lot of information, but you can imagine it almost like a bowl of hummus or ground up beans here, right? So you can certainly measure the level of spiciness or the different flavor profiles in there, but it doesn't tell you where in the tissue those changes might be occurring to contribute to the overall bulk pattern that you're seeing. Now, an advance of that has been the single cell resolution RNA-seq approaches, where the tissue can be dissociated into individual cells and the expression can be measured in each one of those cells. Now, you can compare this to, say, a bowl of bean salad here. So here you can see individual cells and you can measure their taste and flavor and profile individually, but you don't know how they were arranged spatially with respect to each other in the original tissue. And the disruption itself to cause that single cell suspension you can lose rare cell types. Some cell types, like podocytes, for example, don't extract well from the tissue because they're so large and complex. And so you can skew and bias the types of information you get just by doing the procedure in the measurement. So this is where spatial transcriptomics really is powerful, because not only can you measure the level of expression of the individual cells in the tissue, you can actually localize them and their relative positioning with respect to each other. And all of this is done really with minimal disruption to the tissue. It's intact, it's been fixed, and it's been sectioned. And so this is really the real driving force for these spatial transcriptomic approaches. So there are two main platforms here you might hear about. I won't really go into the detail given the time here today, but you'll hear different papers in the literature using these various platforms. They're conceptually different. They both give very valuable information, but they both have their strengths and advantages. The first is the 10x Genomics produced Visium platform. This uses kind of a matrix of spots that capture the RNA that is present on the tissue right above it. And so you kind of get this grid-like pattern. And then this false color coding is indicating the level of expression of a gene, either high or low, localized to a particular area of the tissue. So it's very useful for getting kind of a geographical view of the entire tissue section. The nanostring-produced GeoMix DSP, or digital spatial profiling platform, is the one that I'll talk more about today. We have adopted it because it works right out of the box on FFPE tissues like our kidney biopsies with no optimization required. It uses a standard glass microscope slide, so we can receive slides from other investigators and process them for this technology without any issues. And most powerfully for glomerular type diseases, we can precisely outline a glomerulus and capture its signature, which is not possible using the Visium platform. So this is kind of schematically shown here. So if we have a section of kidney that is shown on the left here, this is stained with the PAS stain, which is kind of this pink and purple stain. You can see 3 glomeruli over here. So using the Visium approach, it would overlay a matrix of capture spots, and you can't control where those spots actually land with respect to the tissue. So some of these spots, and this is really shown to scale for this technology here, will land wholly within a glomerulus, and others will be outside of the glomerular structures. But some of them may straddle the boundary between a glomerulus and the tubulointerstitium. And so the gene expression profile that you get is going to be a composite of this complex mixture of cell types that are within it. Now, in the geomics approach, here we can precisely outline each one of these structures. And so sure, it will be a multicellular region of interest, or ROI, but we know exactly where we place that region. So we have better confidence in our expectation of what cell types might be in there. So again, this is the reason why we chose the geomics workflow, because especially for glomerular disease, the power of being able to precisely define the structures. Now, we can also define whether these structures were pathologically affected or not. So do they have FSGS? Do they have a crescent or not within a given biopsy? And that was really the power of the technology that we wanted to explore. So let me skip over these slides here for time. So just as a preview, even though this technology only came out in late 2019, 2020, really, it's really being taken off. And even though these are expensive methodologies, the power of the information that is coming out from them is exciting people to use this in a variety of different contexts, not just in the kidney, of course. So let's dive into kind of some of the challenges and the opportunities. So hopefully now you have a conceptual idea, a framework, what I mean by spatial transcriptomics, how we can localize gene expression within a tissue and assign it to a particular histologic structure or appearance. And so let's think about some of the opportunities. Can we actually use this for clinical purposes? Okay. But before we can get there, I wanted to show you some vignettes about how there are both challenges and opportunities for doing this. So first, let's consider how we can identify the different gene expression patterns of histologically different looking glomeruli and see if we can get it to work on kidney tissue itself. So that's the first opportunity, is to discover new disease mechanisms and understand the heterogeneity of disease, both within a patient and also across and between patients. So for this, our first study using this technology was on collapsing glomerulopathy. And many of you might be familiar with this. So this is an aggressive glomerular disease process where instead of having normal patent glomeruli and open capillary loops, the capillary loops will kind of collapse and scrunch down. They will not be patent. And you will often see a proliferation or reactive appearance of the overlying epithelial cells, some of which may even contain protein droplets within their cytoplasm. So this is usually also associated with marked decline in kidney function, heavy proteinuria, and progressive kidney disease, often progressing to end-stage kidney disease and requiring replacement therapy. Now, this was originally described during the HIV epidemic in the 80s. It was not originally thought that it was associated with HIV, and it was described separately. But then smart investigators found that there was this close association. And since then, several other risk factors have been associated with collapsing glomerulopathy, such as high-risk APOL1 genotypes, bisphosphonate exposure, other viral infections, notably SARS-CoV-2, and we had a study published on this as well. And so there are many, many triggers potentially that can cause this a disease process. Now, some of the challenges of studying collapsing glomerulopathy is that not all the glomeruli within a patient's kidney biopsy may have this histologic appearance. But even having one glomerulus that looks like this in the right clinical context is sufficient for this diagnosis, which of course is a serious diagnosis to be giving to the patient. And so it has been challenging to study this because of the heterogeneity of the disease process, even within the tissue of a given patient. A previous study had used laser capture microdissection so they can pluck out the individual glomeruli to study their expression signature. But because of the poor sensitivity of that technique, they needed to pool a lot of glomeruli, both normal and abnormal. And so that dilutes the potential unique signature of the diseased glomeruli compared to their non-affected counterparts. And so we thought that this was an ideal scenario to explore whether spatial transcriptomic technology could give us new insights into understanding this disease process. And so we did this exactly. So this is a study we published early last year. We could label the tissues, we can extract individual glomeruli, and in this heat map is showing high expression of any given gene as a red-orange color and low expression as a blue color. Each individual column that you're seeing represents the expression profile of an individual glomerulus taken from a 5-micron thick kidney biopsy section that was taken from the routine clinical material. Okay, so right out of the box, cut a section, did the assay, and we could get this information for each individual glomerulus. So we did this with from 3 patients who were normal controls, 3 who had HIV disease, and 3 with COVID-associated collapsing glomerulopathy, and profiled about 3 to 6 glomeruli from each of these individuals. And we could profile over 1,800 genes in this original iteration of the technology. Now we can do the entire transcriptome, 18,000 genes. But already from this, we can see that there are distinct patterns, both between the normal and the patients with disease, and even between the patients and different disease processes themselves. We can reduce this complex dimensional information into a principal component plot, which is shown on the right over here. So this is a way to basically take that 1,800 gene information for each glomerulus and reduce it into a single point in 2-dimensional space. So here you can see that the glomeruli from the normal controls were pretty tightly clustered and very similar to each other in their overall expression profile. And you can kind of see that by just their color coding over here of the normal columns. But by contrast, the glomeruli from HIV, patients with HIV disease and COVID disease were quite distinct and spread out in their expression signatures. And this already told us that this was going to be an important tool for us to study this disease. So remember again, for the clinical context, all of these patients would have gotten the same diagnosis. They would have gotten the pathology, on the pathology report would have said collapsing glomerulopathy. But we can see that there are actually very different kind of expression signatures that are underlying that same histologic appearance. So we can take this further and then ask, in a given for a given histologic appearance, is the glomerulus collapsing, or does it look more normal, regardless of where it came from, normal or patients with disease? We can ask, what are the differential gene expression programs? We can also ask, in the patients with HIV versus COVID disease, are there different mechanisms at play? And again, the individual genes are not important here for this talk, but we can see that we can, by having that additional level of spatial annotation and the disease context and additional metadata, we can ask almost any one of these questions and get insights into the mechanisms and pathways that are underlying these processes and these appearances. And this is many, many more genes that than were published in the previous study because we didn't have to pool the glomeruli together. We could study each glomerulus individually. Now, I mentioned to you that these are multicellular regions that we're selecting, the entire glomerulus. So it's going to be composed of podocytes, mesangial cells, endothelial cells, perhaps even infiltrating immune cells. So can we get some information about which particular cell types might be involved in these disease processes? Could it be that, in HIV versus COVID disease, that the podocyte or the endothelial cell are differentially affected, and that's causing the difference in expression patterns that we're seeing? So one approach to do that is to use single cell RNA-seq data. So that is, again, when the cells are all dissociated and we have their expression profiles. We can then use that to estimate the relative composition and state of the cells in a multicellular structure like an entire glomerulus. Okay? So an example of this is shown here, again, with that collapsing glomerulopathy data. So we can look at the three major cell types here, the podocyte, mesangial cell, and endothelial cell in the glomerulus. Here, the yellow color means high or normal level of expression. Black and purple means low levels. So focus here on the normal controls here, and we can see that they have high expression of podocyte, mesangial cell, and endothelial genes in the glomerular regions that we selected. By contrast, in patients with COVID disease and HIV disease, these are getting dimmer, but they're not uniform. So for example, in this COVID patient with COVID-19, the podocyte signature was retained across the 6 glomeruli, but there was a loss of the endothelial cell signature. Okay? By contrast, in these patients with HIV, they were retaining many of the signatures of these 3 major cell types, but perhaps gaining abnormal signatures, such as that of parietal epithelial cells within the glomerulus. Okay, so this is again a way to allow us to think about which cell types might be contributing to the particular pathology in collapsing glomerulopathy. So this has now really informed the way we think about collapsing glomerulopathy. These studies and a whole bunch of other literature as well, describing collapsing glomerulopathy in different contexts. So I think this is really the way we think about it now, is that you can either severely injure the podocyte or the endothelial cell separately, and both of those two injury processes can lead to a collapsing glomerulopathy. So we typically think of collapsing FSGS as a kind of a podocyte injury process, and that's true in the context of, say, high APOL1 induction with the high-risk genotypes, and other toxic insults such as pomidronate. But it is also true that blocking or injuring the endothelial cells, such as with anti-VEGF therapies, again, APOL1 high-risk genotypes and ischemia, can also lead to this disease process. And so these two have different kind of expression signatures that we can actually measure in the kidney biopsies, and is informing how we approach and talk with our nephrologists about this, because there might be different interventions that could be used in these contexts. Another way to think about this is that in a biopsy represents a single sample, a single snapshot of the disease process in a patient. And we typically don't go back to get a repeat biopsy to see the progression or outcome or resolution, hopefully, of the disease. But hopefully, if we have samples from multiple patients, we can try to infer the natural history of a disease process. And this is what is known as the pseudotime progression. It may also tell us whether there are different paths of injury in a particular disease process. So we applied the same type of analysis to our cohort of patients with COVID and HIV disease. And we could see that there were very different trajectories coming from the normal glomeruli, where the HIV would have a strong interferon-inducible pathway, versus COVID in 2 different flavors had 2 different distinct disease trajectories or phenotypes, if you will. And we can overlay on these trajectories the expression of any given gene, for example, I5-6, which is interferon-inducible, showing that it is contributing to this distinct appearance and morphology of the glomeruli in patients with HIV disease. So I've shown you through that first vignette that this is a very powerful technology that can distinguish between lesional and non-lesional structures, mine into their underlying expression pattern. We can understand disease mechanisms. We can even maybe identify different pathways of disease. And all of this is done on clinical samples with a single slice of tissue using what was already available in the clinic. And therefore, since they were not collected with any special precautions, just the routine clinical biopsy, they're fairly tolerant already to some of the pre-analytic variables that might be considered. So let's now think about some additional opportunities, such as maybe identifying subgroups of patients within a particular disease entity. So for this, let's consider minimal change disease. So this is another nephrotic syndrome type of presentation for patients, most common cause in kids. And it's called minimal change disease because by light microscopy, the glomeruli look basically normal. Open capillary loops, normal mesangial regions, no inflammation. However, by EM, by electron microscopy, we'll see extensive or diffuse effacement of podocyte foot processes, which is seen on the outer aspect of this glomerular capillary loop. So this is the defining feature of minimal change disease. We can also see this in FSGS patients, so we really need to rule out that there isn't FSGS to give a diagnosis of minimal change disease. Now, some patients will respond to steroid therapies, others will be steroid dependent, and others will be even steroid resistant. Again, so they have different prognostic implications, but yet they get the same pathology report saying minimal change disease. So this was, again, something we thought was an opportunity to try to understand whether we could stratify patients better using the information embedded within the kidney biopsy tissue itself. So we're not unique in doing this. So this is a group from Michigan, Matthias Kretzler and Laura Mariani. So they had a large cohort, again, across multiple continents even, where they pooled FSGS and minimal change disease biopsies. And to do this, they actually took a prospective biopsy core that they under a microscope, micro dissected out the glomeruli versus the tubulointerstitium for RNA-seq analysis, so bulk expression analysis. So again, they had to pool a lot of glomeruli, and they had to take a separate biopsy core rather than the actual clinical specimen. Regardless, when they looked at this in both FSGS and minimal change disease, by looking at the expression signature, they were able to identify 3 clusters of patients. And most importantly, this cluster 3, shown in the red line, had a poor outcome compared to clusters 1 and 2. So they wondered what was driving this, and then they found that this cohort of cluster 3 had a high signature for TNF, tumor necrosis factor. And they could actually measure this even in the urine as kind of a proxy of doing the kidney biopsy to inform and say that these patients may have a more aggressive disease course. Note that most of the cluster 3 patients with the aggressive disease course belong to the FSGS cohort, and only about 10 to 12% of patients with minimal change disease will belong to this kind of expression signature. So we wondered whether we could do this on a clinical biopsy rather than having a prospective biopsy collected just for microdissection. And in fact, we were able to do this. So this is now a study of about 14 patients with minimal change disease. We were able to see 2 distinct subgroups. We didn't capture this cluster 3, probably because of the low frequency of this expression signature in patients with minimal change disease. But regardless, we were able to capture the signature of cluster 1 and 2, and we were able even to see what are the differential genes that are expressed that's driving that signature. And I think the most impactful point for me was that we could do this again with a clinical biopsy. Okay, so you can imagine a scenario where the patient has nephrotic syndrome. They're trying to figure out, does this patient have FSGS or minimal change disease? Biopsy is done. From that biopsy, we could derive an expression signature and say this patient is in cluster 1 or 2, probably a good prognosis, versus whether they are in cluster 3 and may have a more aggressive disease course. So that was really what excited us from the results of the study. But furthermore, we noticed that there's even heterogeneity even within the patient. So while given patients' glomeruli, each individual column here may generally fall into group A or group B, some patients actually straddle it, okay? And so this is telling us that we may actually have fine gradations of disease even within a patient, and it may not be as simple as cluster 1, 2, or 3. And we don't have an answer for that complexity yet. So this is one of the challenges in front of us. But I think this is showing you very clearly that this technology can allow us to identify subgroups of patients and maybe even inform how we diagnose and think about their outcome and prognosis. So one challenge for thinking about clinical translational aspects of new technologies is to think about their rigor and their reproducibility. How reproducible is this test going to be if we do it today versus a year from now versus 5 years from now? And so to address this, we devised an experiment where we had kidney nephrectomy tissues from 4 different donors, plus one section of the kidney tumor for the reason why these kidneys were being taken out in the first place. And then we selected more or less the same regions that we could in 4 consecutive sections of this tissue block, okay? So the idea here was, if we do this assay on 4 consecutive days, try to capture the same region as much as we can, do we get the same results? So how technically reproducible is this platform? And so we can do this, and again, we can see in a principal component plot that we can nicely distinguish the kidney tumors from the medulla, from the cortex, and the glomeruli. And if we ask how much of the variability in gene expression is due to the different histology, meaning glomerulus versus tubules versus cancer, we can see that almost 85% of the variability in gene expression is because we can identify different histologic structures. So that's expected and that's good. There was also an additional component of variability that came from donor to donor. So the glomeruli from donor 1 versus donor 2 was slightly different from each other. There was also some variability introduced by the ROI selection. That could be because of our manual subjectivity in drawing the regions, or also because the structure that we're selecting is actually changing as we go through one section to the next. You can imagine a glomerulus coming in and out of a plane of section in a particular slide. But all said, these are all biological forms of variability that we kind of expected to see in the experiment. But the technical day-to-day variability of the platform was only about 2% in our hands. So this gave us real confidence and excitement that this could form the basis of a clinical test because it is very robust and reproducible. So now let's consider some challenges and think about, if we want to do these kinds of studies, we have to profile a lot of patients. As I mentioned to you before, biopsies are single points in time. So to understand the spectrum of the disease, we need to capture enough of the disease pathology spectrum and therefore its gene expression signature therein to try to understand how we can impact clinical management and outcomes. And so for this, I want to actually learn, and we are learning from our colleagues in Edmonton who have been doing this for much longer than we have in the transplant scenario. So for here, I'm referring to the molecular microscope diagnostic platform, where again, a separate core of kidney tissue is submitted to the laboratory, and they grind the whole thing up and extract a composite gene expression signature from that piece of tissue. And by doing this prospectively over almost 20 years now, they've accumulated data from over 1,700 biopsies. And there they can again place all of that information onto these principal component plots. And they can see groups which have T-cell mediated rejection phenotypes, acute tubular injury phenotypes, infection phenotypes, or antibody mediated rejection phenotypes. And so then they can place your patient's biopsy into this high dimensional space and say, well, your patient has scores that are high for, injury and rejection. And so maybe this patient has antibody mediated rejection. And then this is used as an adjunct to the pathology diagnosis. You can imagine if there's some equivocal findings on the biopsy, this may be the deciding factor to say, okay, well, let's go with antibody-mediated rejection and treat the patient as such. So this is an example of how they were able to translate a molecular expression technology to the clinic, but they required it to have a stable platform. They required a separate piece of tissue to be submitted, and they could not distinguish what the components in the tissue were. And so I think the new technologies will allow us to leapfrog this in a way because we can very rapidly go to our archival tissues and get up to 1,000 biopsies in short fashion. And we can use existing clinical material such as FFPE tissue blocks to do this without requiring a separate core of tissue. And so learning from their experience, I think we can rapidly progress our application of spatial transcriptomic technologies to a whole variety of kidney diseases beyond even glomerular disease processes. So for this, we would have to have benchmarks that we can then run at different intervals to make sure that we're getting the same kind of information from our reference block so that we can then normalize any new patient material against the single large database that we are generating. Some power considerations as well. We need to probably profile hundreds of patient biopsies to really get good discrimination and power to distinguish. Because if a disease or signature is prevalent only at a low percent, like I showed you for cluster 3 in that minimal change disease, we would really need to profile hundreds of patients to be confident that we're capturing a sufficient number of those patients and the signature that they're in. So these are some of the considerations we're thinking about as we design these studies, which is a challenge because some of these technologies are expensive as well. So how can we get up to these kinds of numbers? So finally, we can think about validation as well. So these are all done at the gene expression level. Does it actually then translate into a more simpler clinical test, such as immunohistochemistry? And we found in our previous study, for example, that these two genes, PAX8 and MME, were dysregulated in collapsing glomerulopathy. This one going higher in expression, this one going lower in expression. And so you can see in a normal glomerulus, there is not much PAX8. There is a lot of MME. And in collapsing glomerulopathy related to COVID or HIV, you can see increased brown signal over the glomeruli for PAX8 and decreased MME signal. So this was following the prediction that we got from the gene expression signature. However, we are also flipping the script and we can ask, having found a biology, can we use spatial profiling as a validation tool? And so this is a recent paper that just came out just this week from our laboratory. We also do a lot of other types of genomic studies. So we were trying to, in the study, we were trying to understand the differences between the kidney cortex and the kidney medulla. And we cued in on this transcription factor, POU3F3, which seemed to be playing an important role in the kidney medulla. This is a transcription factor which is very important for distal nephron development. So the knockout of this gene in mouse kidney causes a failure of the collecting duct and distal nephron to form. In the human fetal kidney, this is also expressed by the black dots, POU3F3, in the cells that were in the metanephric mesenchyme and also in the ureteric bud. So really those components of the collecting duct and the distal nephron that have to hook up together properly for the collecting system. But no one had explored its expression in the adult kidney medulla, and our data was suggesting that it was important there. So to validate this, we did a spatial profiling experiment where we took a section of kidney tissue and again outlined different structures like glomeruli, kind of distal nephrons in the cortex, proximal tubules, medullary rays, and explored the gene expression of 18,000 genes within there. And here's the sections in the medulla as well. And we could see, again, using the cellular deconvolution approaches, that each of these structures that we had outlined histologically contained the expected cell type. So that was the first control. So our glomerular regions contained podocytes, mesangial cells, and glomerular endothelial cells. In contrast, our outer medullary inner stripe sections contained medullary TAL cells, principal and intercalated cells as well. So this gave us confidence that what we had selected were what we really thought they were supposed to be. And then from within there, we could ask what was the expression of POU3F3. And sure enough, it showed very low expression in the glomeruli and proximal tubules, and kind of a stepwise progression of expression into the medulla, kind of what we had predicted from our other genomic studies as well. So this was kind of a reversal of the approach, using spatial profiling as a validation tool. So of course, we're helping as a director of the core facility at the University of Washington, we're helping many other labs do projects using spatial profiling in kidney and beyond, and a variety of tumors as well. We're getting a lot of information now how we can use this technology to develop new insights, which can be very rapidly used in the immunohistochemistry type of mode in today's practice. But also we can think about how we could use a composite gene signature in the future sometime to inform diagnosis and stratification of patient prognosis as well. So if you are interested, we are happy to collaborate with you as well on your projects. We have a lot of experience with a lot of different tissues and also designing the experiments as well. And I do this together with my colleague, Kelly Smith, who's also a renal pathologist. So let me just quickly summarize some of the key points of the talk, and then I'll just, this was an already kind of exciting and mind-blowing. I'll just preview some of the future technologies that are coming as well. So I've shown you that this technology is both rigorous and shows high reproducibility with low technical noise. So it's encouraging that it could be used in a clinical environment. It allows us very clearly to identify individual structures, get their gene expression, and correlate it to the histology. So collapsing or non-collapsing appearance, for example. And so this approach gives us incredible power to study both intra-patient and inter-patient variability, especially if we can profile multiple individuals. And that's the challenge. We need to really think about profiling lots of them uniformly. But the good thing is that this is tissue sparing. We only need one 5-micron slice from an existing archival tissue block of FFPE. And so we can really think about applying this broadly at multiple sites to collect the data from this type of technology. And ultimately, we hope that, if we can translate this into the clinic, and there is precedent, such as in the MMDX scenario for kidney transplant, it will improve our understanding of the complexity of kidney disease and also the complexity of our patients and the heterogeneity and variability, so that we don't have to treat them all exactly the same way because they may not respond in the same way. And if you were wondering about how much it costs, it's roughly about $1,000, $800 to $1,000 per kidney biopsy to get this kind of information. So compare that to, say, cell-free DNA assays, which are about $3,000. And even the MMDX is probably in the same range or a little bit more. So we're not talking about an outrageous sum here to extract this kind of information with existing material. So just briefly in the last minute or two, I just wanted to say that we're not stopping there. There are many new technologies that are coming onto the scene as well. So I told you that these current spatial technologies that are more widely deployed are multicellular. So we have to outline an entire glomerulus. And then we have to use complex mathematical tools to try to understand which cell type might be affected within there. However, newer technologies are allowing us to get this kind of gene expression information at the single cell level. And so we can actually label a tissue with different antibody markers, segment them or outline different individual cells, and then measure the expression of different genes as these measured by these little dots within the tissue. So just to outline that, here's a labeling of a tissue. We can use different antibody labels to identify different structures. We can then use those labels to outline individual cells and then measure the gene expression within there. And from there, we can identify individual cell types and groupings of cells that can form neighborhoods. So we can then understand what are the receptor and ligand interactions between neighboring cells. Are there clusters or certain relationships of cells that go together? For example, do we see a lot of plasma cells around a particular tubule? Does that mean something for that disease? So these are kind of where some of these technologies are coming onto the horizon, and we're trying to explore them as well, mostly in kind of a discovery mode, not necessarily in a clinical translational mode just yet. So finally, I'll stop there. I have a lot of people to thank from our core facility and funding sources as well, and our collaborators. And if you're interested in some of the things that we do even beyond this, I encourage you to kind of take a look at that QR code and that website link. And I'll stop here, and I think we have a few minutes for questions as well.
Yeah, we do. Thank you so much for an amazing tour through what is and what can be possible with tissue biopsies. If you want to stop sharing, and I think I can see a tiny little bit better, everybody. So one of the questions from Dr. Matzel is whether technology has been looked at temporal gene expression in normal abnormal kidney development. So Doug is a pediatric nephrologist, which is why he's asking that question. But
yeah, a great question, Doug. Thank you. It hasn't been, at least in a published form, but clearly you can think of any question in kidney disease and biology, and this would have application. So some of the applications that I have seen, for example, for the Visium platform from Ben Humphrey's group at WashU, they have looked at a mouse model of acute kidney injury where they ligate the ureter of one side and then use the other kidney as a control. And that also has shown temporal changes as a progression of kidney injury, and also sexual dimorphism. So there are big male and female differences in kidney injury response. So you can imagine that same kind of approaches could be used during kidney development. And there, some of those map-based expression profiling might be the best way to explore that as well. So great question. Yes, I think Mazi, you had a question?
Mazi, you have your hand up. You can unmute.
I believe you can hear me now.
Yep.
I can, thank you.
Great talk, fascinating work. Thank you so much for taking the time to share it during your holiday. I have 3 questions. Adira can stop me and I can ask.
Go ahead, Mazi.
So the first question I have is about the probes. Ram, how much flexibility do you have? Do you have to lock in a certain set of probes and just go with those in perpetuity, or you have room for improvisation? For example, in your collapsing glomerulopathy paper, could you have asked for probes to look for viral coded material in the biopsy and see how that would distribute, if any?
Yeah, great question. So the probes are sold as sets, but then you can always add custom probes to it. And so that's just a separate design consideration and of course cost. So in that paper, we actually did include a custom probe set, which because the initial one had only about 1,800 genes. So we added another 25 or so genes related to COVID biology. So we included like ACE2, and we also included probes to detect SARS-CoV-2. Because there was a big debate about whether there was direct infection in the kidney versus not. And in that study, we did not see any viral transcripts in any of the tissues that we profiled, which has been actually corroborated in other studies as well using this technology. So to answer your question, there is flexibility on the probes, and you can even add in probes targeting non-human transcripts or RNAs in the process.
Perfect.
So, before you ask your next question, Sean also has a hand up. Can I do a little switch and bait? Okay, Sean.
Hi, Sean.
Hi, thank you. That was a great talk, and I actually managed to follow some of the translational talk, which is always an indicator of a great talk. So I guess my question is just an extension of the Neptune results, where they looked at minimal change and FSGS and outcome. Minimal change is very homogeneous when you look at it, so it may not take advantage of some of the being able to narrow in on affected glomeruli, whereas FSGS, you can identify the lesion or the glomeruli with the segmental sclerosis lesions. And in terms of outcomes, usually, at least for patients that we think of immune-mediated podocytopathy driving their FSGS, their first treatment would tend to be steroids. So, you know, is there a possibility of correlating this with clinical outcomes, but narrowing it down to perhaps things like steroid resistance or steroid sensitivity in FSGS, which generally would be associated with long-term outcome and might inform at least the first-line therapies for patients, and then potentially avoid using steroids in patients who are unlikely to actually respond and avoid toxicity?
Yeah, great question, Sean. Lots to unpack there. And I would say actually, and I went over that quickly, but when I showed the heat map of our own experience with the minimal change disease cohort, even though we think of minimal change disease as being uniform and homogeneous, and that was mostly the case, some of the glomeruli, even within a given patient, actually had disparate assignments to cluster A and cluster B. So we're trying to understand what that means. Is it an 80-20? You know, what's the proportion? What does that look like? And so to do that, really, we are now building out larger studies, doing exactly what you're describing, right? Where we can profile hundreds of patients where we have clinical outcome data Did they respond to steroids? Did they not? Did they eventually develop FSGS on a separate biopsy? For example, are they associated with anti-nephrin antibodies, which have been implicated in minimal change disease? Are there different pathways that may be at play when you have those antibodies versus not? All of those questions are exactly what we're interested in deploying using this technology. So it may be that in the future, we don't use this technology per se to identify a particular disease, patient cohort, but it may inform a particular biomarker, such as in the TNF signature in the Neptune group, or it may inform a particular panel of antibody stains that we do on the tissue biopsy itself to say that this patient is going to be steroid sensitive or resistant or responsive to something else. So you're thinking exactly along the lines of what we're planning to do.
Yeah, I think this
Great.
yeah, thank you. One last short question, commenting on spatial transcriptomics, how it might complement the other omic strategies like gut microbiota genomics and metabolic proteomic profiling.
Yeah, so that's a great question. So KPMP, this is really the driving focus where they're looking at human biopsy tissues and using all of these omic technologies. Both the dissociative ones where they separate the tissue into component cells and also the ones which can utilize a tissue section. They're looking, of course, at gene expression, which is the easiest to measure across the entire genome, but we can't ignore proteins, we can't ignore lipids and other metabolites. The resolution for those technologies is not quite single cell, but they're getting there very rapidly. So I think the challenge for KPMP and the field in general is going to be, how do you integrate all of these together, right? How do you take one data point of one technology, correlate it to the same region in a different technology, and then integrate that with the clinical data? That's really going to be the challenge, I think, of this next phase of investigation in this field.
Yeah, no, I think everyone's like very excited about all of this technology. You and I were talking before sort of about the amazing plenaries at the ASN that also opened up our eyes to technologies and knowledge about how bacteria talk to each other and sense each other, and taking all of these new tools with thoughtful application to the human condition. It's quite an exciting time in nephrology. And then obviously the correlates of not just describing the appropriate responses, non-responses, but then they're targeting new therapies, understanding the biology so much better is going to be really exciting. So again, just because we're out of time, I just want to thank you for a really inspiring and terrific talk. It's always a pleasure to hear you. You have a wonderful way of simplifying very complex things to make us all believe that we understood this, at least for the hour, but hopefully helps us really understand and appreciate the value of biopsies. No matter what tools, they're all getting better, and whatever the sophistication we have. But I think certainly we're not that far away, and I know that the pathology group here is interested in working collaboratively across the different technologies.
Of course, happy to do that.
Yeah, thank you so much, and thanks everyone.
Thank you all. Thank you all for your
for
time.
your Holiday Friday talk. Thank you.
No problem. Thank you.